It takes 6 tons of rock to produce just 1 ounce of gold. Miners work 6 days a week, digging, crushing, and grinding the ore to unlock the gold.
If you grind it fine enough, you're going to release the gold. It's not in the grain boundaries or anything like that. It's actually free gold. If you look at this table here, it's nothing more than an automated gold pan. It's doing the same thing the old timers did in the stream, except it's much more efficient and it's doing a lot faster and a lot more material. See that little golden streak coming down the table? That's the gold.
That streak is only half the gold in this ore. To extract the rest, the rubble goes to giant tanks of cyanide.
Cyanide dissolves gold just like sugar dissolves in water. It makes a pure solution out of it.
After the gold was extracted, the cyanide solution was dumped into the town's Whitewood Creek, killing everything in the water. For over 100 years, the creek ran black. The Environmental Protection Agency gave Homestake 1 year to develop a plan to get the cyanide out of the creek. The mine managers weren't sure how to tackle the problem and keep the mine profitable. For the people in Lead, South Dakota, profits for Homestake meant jobs for the town.
There's very few things besides cyanide that will dissolve gold, and they're a lot nastier than cyanide.
Homestake's chief executive officer added a challenge: make the stream pure enough for trout. Homestake hired a local biochemist, Jim Whitlock.
The problem was not money. They were willing to spend whatever it took, but the problem was there was no available technology to do this. There wasn't anything that they could take off the shelf. The fate of the mine, everything was riding on it.
Whitlock and his team decided to use their background in microbiology to try something new.
We started with a simple knowledge that bacteria could tolerate cyanide, and we knew that some bacteria could tolerate a lot more cyanide than others. In fact, this was a standard means for microbiologists to identify bacteria based on how much cyanide they could tolerate. And so we looked into that in depth, and what we found out is that they actually broke the cyanide molecule into 2 parts, and so that they could actually use it as a food source.
It worked. Today, all the water from the underground mine is filtered through these tanks, each containing about 20,000 pounds of Pseudomonas bacteria attached to plastic discs.
Now, the bacteria like to attach to things, and they have a little tail that they use to propel themselves around in the water. They'll discard the tail and secrete a slime layer, which is how they attach to the plastic disc. So as the disc rotates very slowly, about 1.5 revolutions per minute, partially submerged in the water tank, the bacteria are attached to the disc. So they're in the water, and then they're back in the air, and then they're in the water. So when they're in the air, this helps satisfy their oxygen requirement. And when they're in the water, then they're actually picking up cyanide and absorbing metals.
Helicobacter pylori has affected more than just ulcer disease. Now scientists are studying the link between bacteria and many chronic illnesses like asthma, periodontal disease, hardening of the arteries, Crohn's disease, colitis, rheumatoid arthritis, and even some cancers.
If even one of these diseases proves to be caused by bacteria, and thus goes from the incurable or where the cure is a very radical and unacceptable to many patients, take it off that list and put it on the list of diseases that can be readily cured by antibiotics, that would be a major contribution.
This is the Institute for Genomic Research, or TIGER. Here they pull out a bacterium's genetic blueprint, its DNA, and break it into chunks that a supercomputer pieces together. If the whole DNA is the book describing a bacterium, the genes are the pages. The computers put the pages in the right order.
I mean, what we actually see is a couple million letters of A, C, Gs, and Ts. I mean, that's the genetic code. That's what we determine when we actually determine the sequence of the genome, is the order of the letters. The real task is in interpreting what that means. You know, what words, what sentences, what messages are there. And we're finding some phenomenal messages hidden in there. The things that are pre-programmed into these genomes is, I mean, it's absolutely amazing. In each one of these genomes that we do, it increases our ignorance quotient quite substantially.
Once the genetic blueprints of microorganisms are completed, the functions of the genes are studied. As researchers understand which genes are responsible for growth or disease, they can be manipulated for many purposes. And it's given us insight into a much larger question.
We like to think that we're highly evolved, and therefore our chromosomes are probably highly evolved. And bacteria are primitive. Well, in fact, the opposite is true. The bacterial chromosomes are very highly evolved. If you think of organisms that replicate every 20 minutes to an hour, and they've been doing that for billions of years, they've gotten very efficient at everything they do. So there's no wasted letters in their alphabet. Essentially, they're wall-to-wall genes for all practical purposes.
Understanding the genes of microorganisms also opens up thousands of new targets for antibiotics, vaccines, and industrial applications.
It's amazing the diversity that's out there. We know a tiny fraction of 1% of what's out there. And the smartest thing we could do in this research is, you know, go try and find new ways to culture thousands and thousands of new microorganisms and find out their genetic code and find out what's there. It's going to revolutionize virtually everything we do.
Yellowstone National Park is one of the richest places on Earth for all microbial life. Bacteria thrive in the extreme conditions found in the pools. Tourists see the spectacular scenery. Researchers come to understand how life can survive.
Many of the organisms that live in these pools have to eat. So those are called heterotrophs. So they have to consume things in their environment. And in order for a microbe to consume something in its environment, it has to make an enzyme that can break that particular item down. Well, if we see a piece of wood in this hot pool and we pull it out and we see that the outside of that piece of wood is bleached white, you automatically ask the question, how did that happen? So when we look at the microbes associated with that wood, studies have shown that these microbes have that capacity because we've cloned the genes from these organisms that encode those particular enzymes and have found that, in fact, they are adapted to breaking down these wood fibers under these extreme conditions.
By cloning the genes that produce whitening enzymes, they can turn it out in quantity. That means paper companies can use bacterial enzymes to whiten paper and lessen the need for chlorine. It's good for the environment, and it's big business. Enzyme production is an $11.5 billion a year industry. Most antibiotics come from bacteria living in the soil. As the search for new antibiotics continues, scientists are learning just how little we know about the microbial life beneath our feet. The search for new microbial life has taken researchers from backyard soil to the ocean floor to the heavens. A meteorite from Mars has fossilized structures that look suspiciously like small bacteria. Scientists are coming to the conclusion that life may be far more common in the universe than we had supposed.
Anywhere that there is liquid water, there is a possibility of life, and we know that here on Earth that's just a fundamental requirement. And in fact, anywhere on Earth where there is liquid water, you find there is life. And so the fact that on planets such as on Mars, where you might have hydrothermal features under some of the ice flows that might make liquid water is very exciting.
The experts say this is the beginning of the golden age of microbiology. Industry, medicine, the definition of life itself are all being changed by single-celled creatures you and I can't even see.
Everyone should look at pond water at least a few times a year if you're a microbiologist, because now the days of technology and DNA and and high-tech things, it is nice to go look in a microscope every once in a while and look at that life and that diversity of life, because you can't just put it in a vacuum or look at it as a bunch of A's, T's, G's, and C's and a chromosome. This is not life in itself. So you should go look at pond water. Everyone should look at pond water, and they'll understand in that instant is more about a feeling about what life is than almost any other kind of experience they could have.